Alright guys, so watch and listen carefully on these. Okay, they're going to be just 1 mark each. They're either right or wrong. Okay, so our first one here, we've got an ionic compound containing sodium and a complex ion or polyatomic ion carbonate. So this stuff is sodium carbonate. That is not a capital S, by the way. It just looks that way. Okay, I'll make it a little bit smaller. Now, today, I'll let capital letters go. Next time, I won't. Okay? So, if they got capital letters, just write beside it, no capitals. But I'll let it go today. Okay? Next time, not so much. Alright, next one here, we've got iron, and we've got sulfur. Now, the problem with this is that iron has more than one possible charge, which means its name is going to need what in it? Roman numerals. Okay, so this is going to be iron sulfide, but we've got to figure out which iron it is. So we go back and we do this. We say, all right, we look at the non-metal. Sulfide is always -2. Since there's only one iron here, it must be +2. So this is iron 2 sulfide. All right, number 3 is a molecular compound because they're both non-metals. So this name is going to require some prefixes. So we're going to be looking at triphosphorus, okay, pentabromide. Number 4, I've got copper and I've got a complex ion sulfate. Does copper have more than one possible charge? Yes, it does. So it's also going to need a Roman numeral. Sulfate's always -2, so copper must be +2. So this is copper, Roman numeral 2, sulfate. And for number 5, we've got a molecular compound because they're both non-metals. So we're looking at carbon, okay, tetrahydride. Okay, questions on any of those? All right, remember they're 1 mark each. There's no half marks here, guys. They either got it right or they got it wrong. Okay, so now we got to write the formulas. Number 6 is a molecular compound. To figure out the numbers, I just look at the prefixes. Dinitrogen means N2. Hexachloride means Cl6. Okay, and they do have to leave it that way. You do not reduce molecular compounds, you only reduce ionic compounds. If the prefixes tell you it's 2 and 6, it's 2 and 6. Okay, for number 7, we've got cobalt 2 fluoride. It's ionic. Okay, that's calcium, not cobalt. So, cobalt 2 plus, fluoride 1 minus. Okay, now I drop and swap. So the -1, or just the 1, comes over here. We don't write that. The 2 goes over here. So we're looking at COF2 for that one. Okay, for number 8, we've got 2 complex ions. Okay, does that change the way we do this at all? Not really. Okay, so we write ammonium, NH4, and we write phosphide, PO4. Okay, now we have to drop and swap because they're ions. Okay, this one is -3, right? I gotta double check. I'm forgetting. Phosphate, I'm pretty sure, is -3. Yeah, -3. Okay, so -3 here. This one is +1. All right, so that means when we drop and swap, this needs brackets and a 3 beside it. You're right. Sorry, phosphide. Sorry, guys. My bad. Hey, just going to be a P. But we're still going to need the 3 there. Okay, just not phosphate. It's phosphide. Good catch. Okay, for number 9, calcium nitrate. Okay, is that ionic as well? Yes, it is, because calcium is a metal. All right, so calcium is 2 plus. Okay, nitrate is NO3, and it's -1. All right, so the 1 goes to calcium. We don't write that. If I need another number, I put brackets with the number outside the brackets. Okay, and then our last one here, we've got lead 2. All right, so they're telling us it's the lead with the 2+ charge with permanganate. Okay, that is K-M, whoop, not a little m, big M. Okay, and permanganate is, there should be no K there. Sorry, guys. Gotta get more sleep. MnO4 -1. Okay, so now we got to drop and swap. So the 1 comes down here, but we don't write that. It's a complex ion. So, okay, the 2 goes outside the brackets. We're looking at Pb, MnO4 bracket 2. All right, give them a mark out of 10. Let them see it. Then please put them in the box on the top there on the table, please. Okay, so you've got this sheet in front of you right now. Okay, this is essentially what you'll get every time we'll do a lab. Is you'll get a lab sheet that starts out with, okay, the stuff we talked about yesterday. Okay, the problem that we're investigating, okay, is given several unknown samples, how can we classify them as ionic, molecular, acids, and bases in order to find their identity? All right, remember that your job with the problem, and you may want to write this down, okay, in case you forget, your job with the problem is that you need to restate it in your own words in the form of a question. All right, so you have to ask yourself, what is this question, or what is this problem really saying? What are we trying to do in this lab? Okay, well, in this lab, it seems to me, we're trying to find the identities of unknown compounds using only what? Right, human senses. Essentially, we're trying to find their physical properties, or we're trying to use their physical properties to identify them. So you've got to find some way to put that in the form of a question. Okay, how are you going to do that? All right, that's going to be your job when you're writing out the problem. Okay, put that in the form of a question. Now, for the design, we've got to identify our manipulated, responding, and controlled variables, and most importantly, you need to explain why one thing is the manipulated variable, one thing is the responding, and why the following things need to be controlled. Now, we'll talk a little bit about what those are. I'll give you some kind of big, juicy hints here. Okay? Every time we do your procedure, because you're going to be writing the procedure, okay, that series of tests that we're going to run. Every time we run that series of tests, we're doing it on what? Okay. What's going to be on the front desk? Okay, on the front desk, I'm going to have a whole bunch of what? Beakers. What are they going to be filled with? Different chemicals. Okay? Are you going to run the same set of tests on every one of them? Yes, you are. Okay? I guess I just didn't say that clearly enough the first time. Okay? So, every time you run the series of tests that you're going to write up, you're going to run them on a different chemical. Okay? So, are you kind of changing which chemical you do the tests on each and every time? Okay, that should be a pretty juicy hint for one of the variables then. Okay, you're changing something. There's only one variable that you actively change. Okay, what we're going to be watching for, okay, every time we run those tests on a different chemical, should the results of those tests be at least a little bit different? They should. Okay, and is that what we're watching for? Okay, what variable do we watch for? Responding. Okay, there, I gave that one away. Okay, if you missed it, you'll have to watch the podcast to get it. All right, that's what we're watching for. All right, what kind of things could affect these tests that we should keep the same every single time? All right, so in other words, our controlled variables. What are some things that should be the same every time we do our procedure? Temperature should probably be the same. Okay, it's an environmental factor that affects things. Okay, especially things like solubility and conductivity. Okay, what else? Yeah, size. Okay, we've already said we're going to use the same amount of sample every time, right? We said we're going to fill it about a quarter of the way to a third of the way full with the sample in each dish. It's important we use about the same amount of everything. Otherwise, we might not get proper results. I mean, if solubility is dependent on how much stuff you have in there, you might not be able to get it to dissolve very well if you have too much. All right? Or you might not get enough ions if you use too little, and then your conductivity test might not work. So we want to make sure we're using about the same amount of everything in order to make this a controlled experiment. Okay? Now, I've given you 2. Okay? You guys need to come up with at least 1 more. All right? On your own. Because we always have 3 controlled variables as the minimum, okay, when you're listing your controlled variables. There's only ever going to be 1 manipulated variable, okay, because you can only test one thing at a time, and we've already said what that is, okay. We've already said what the responding variable is, okay. You need to come up with 1 more controlled variable in addition to the 2 we've already talked about, okay, and you need to explain, which we sort of did for the first 2 controlled variables, why they're the controlled variables. You also need to explain your manipulated and responding. Your manipulated is the manipulated variable because what are you doing with it? Okay? The responding variable is responding variable because what's happening with it? Okay? You need to tell me that kind of stuff in your explanation. Right? Does that sort of make sense what we're looking for there? Okay. What 3 words does your hypothesis need to contain? If and then. Okay. The if part, remember, is kind of the premise that we're working on here. Okay? The premise we're working on in this experiment is that everything has its own unique physical properties. Okay? If that's not true, this experiment will not work. And every time we do all those tests, we'll get exactly the same results. Obviously, that's not going to happen. Okay? Everything has its own unique physical properties. That's probably what you need to talk about in the if part of your hypothesis. Like perhaps if that's true. Okay? Everyone with me there? All right? That's the premise we're working on. That's what needs to go in the if part. The and part, what do we do in the and part? Anyone remember? Maybe we should look, because we wrote it down yesterday. Okay? What does the and part of the hypothesis talk about? Okay. If this is true, and what does it talk about, guys? It briefly describes something. What does it briefly describe? What you're doing. Right. It briefly describes the experiment. Okay? So, in the and part, you should briefly describe what you're going to do here. Okay? Maybe mention what tests you're going to run. You don't have to say how to do them, but you should maybe mention these are the physical properties that we're going to look for. Okay? So, tests are run for blah, blah, blah. Okay? For these properties or something along those lines. That's what the and part needs to talk about. Okay? Then, what do we need to be able to have happen? Okay? We've already said we're working on the premise that everything has its own unique properties. We're going to run tests to find those properties. What should happen? If everything has its own properties, what should happen when we're done all those tests? What should we be able to do? Right, and as a result, we should then be able to we should be able to identify the unknowns. Okay? We should get unique results that allow us to identify each of the compounds. Okay? Everybody following a little bit there? It's your first time, so it's tough. That's why I'm kind of laying it out a little bit more for you. You'll find that as the semester goes on, I'm going to wean you off of that a little bit. Okay? All right. The big part of today for you guys is going to be this. Okay? Your experimental design. We talked about that yesterday. There's 4 tests that you're going to run. The observable properties test, okay? The solubility test, the conductivity test, and the pH test in that order. All right? So your procedure needs to talk about what does the person need to do when they enter the lab. Okay? How do they need to do each one of those steps? Okay? Now, that should also include some of the preparation work, okay, that goes on before a lab. So when you first walk into the lab, what is it advisable for you to grab right away? A lab coat and Glasses. Okay? The glasses usually can be found in the drawers of all the lab stations. That's probably something you should mention as the first step in your procedure. Talk about getting the safety equipment on. All right? Then talk about where you can find all of the unknown materials, because you're going to need those, and the other materials you're going to need. Okay? I'll tell you right now, all the materials you're going to need are going to be on the front desk and on a small metal cart next to it. Okay? So your conductivity tester, okay, your spot plate or depression plate, and your pH paper, okay, all of that will be on a metal cart. And you'll be able to grab it off of there. The unknowns will all be on the table. All right? Now, remember we said this needs to be explicitly detailed. That's why it's going to take you a while to write it up. Other little things that you probably will want to mention in your procedure are things that will prevent the accidental contamination of the samples. Okay? Every one of the samples is going to have its own lab scoop in it. Okay? Why is that? Right. I don't want you to mix any of the samples together. Okay? Not because anything will happen. Nothing will happen. But if you contaminate, let's say you bring, a red crystalline solid over into something that's a liquid, well, now you've obviously interfered with the experiment by contaminating the liquid sample. All right, so everything is going to have its own lab scoop, or if it's a liquid, an eyedropper in it, so that make sure, when I'm getting this sample, I'm using that one, and I'm putting it right back in that sample when I'm done. Okay, you don't want to contaminate anything. So that's probably something you'll want to mention as part of the preparation work as well. Once you've told them how to get everything and it's on the lab table, then you can say, all right, we're doing the observable properties test. Here's how you do it. Here's what you look for. Here's what you write down. Okay, make sure it's that specific. Okay, then you'll talk about the solubility test. Okay, and how you do that. How much water do you add? Okay. How much sample did you put in the first place? Right? Do you stir it? Do you put a thermometer in it? All of that kind of stuff is going to be important. Okay. Then talk about how to do the conductivity test. That's probably the easiest one, because it's just put it in there and then rinse it off. Okay. And then for the pH test, also pretty easy. Okay. Put the pH paper in and look at the thing. Record your number. Okay. Again, make sure it explains pretty well what to do. Now, do you need to write all of those steps out 12 times? No. Can you just say, like, repeat steps 1 through however many steps you have for each sample? Yeah, please do that. Okay. Otherwise, it's gonna go on and on and on. Right? It's way easier to just say repeat. And I'm not trying to insult your intelligence by telling you that. I've just actually had people do that. Okay. And it's just gonna save you some work. All right. Then basically, guys, that's as far as you'll get today. Okay. Because you can't do anything else until we actually have our own lab data. Right? So that's as far as you'll get today. And that's probably going to take you the rest of class, okay, realistically. So what you're going to have once we get done explaining here is you'll get your Chromebooks, okay, I'll bring them out, and you'll go to Google Classroom, and you'll find the Properties of Compound Lab, except your guys' is pink, okay, because your class is different color than this one. So you just click on Properties of Compound Lab. You'll see there's 2 other attachments here. There will also be the podcast I'm making right now that'll be going in there, okay, later on today. And whatever I record for instructions tomorrow will also go in here, okay, as part of this assignment. So we'll get a little bit bigger over the next couple days. This here is the Compounds Lab data. You're not going to need that yet, because you're going to get your own. This is for people that aren't here. Okay, so if you're not here, or after tomorrow, if you're not sure about your data and you want to check it against mine, that's also not a bad idea. So it's there for you to use. Okay, and then this is the handout that you already have, the compound chemistry lesson 456 properties of compounds lab. That's the sheet you just got. So if you lose that sheet, this is where you go for a replacement. Okay, so for you guys, you'll simply click on here. Okay, it'll say the same thing it did when we did the Google Classroom practice assignment. Okay, it'll give you this page here, and you'll simply click on the one with your name on it. It'll open up in Google Docs and type away. All right. Now, you're going to be working with a group here. All right. Let's say 4 is the maximum size. No smaller than 3. So 3 or 4. Otherwise, it just gets really congested in the lab. Okay. So groups of 3 or 4. If you don't end up in a group, just let me know and I'll put you with somebody. Okay. So you're basically filling in all this stuff today. Okay. You should get to here. right, down here to the chart. You won't be able to fill in the chart till tomorrow. That's as far as you'll get today. Okay, work with your group, talk it over, talk about how to, write up the variables, write up the hypothesis. It's okay if your hypothesis and the group and your group members' hypotheses are similar. They should hopefully not be exactly the same, but they should be similar, okay, because you're working together. All right, I'll understand that if that's the case. Questions?